We were talking about selecting a clone and screening for the clone that you would want. So blue-white screening is a popular tool, at least some time ago. I'm not sure if people are still doing it that much, but blue-white screening it used to be very popular because it helps you to select for those bacteria that are, number one, ampicillin resistant. That means they've picked up the plasmid. Number two, it gives you a color screening tool to select those bacteria that not only have they picked up the plasmid, but they picked up a plasmid that disrupted the LAC-Z gene because an insert ended up going in there. So here's how this works. The blue-white screening method. So here's your original plasmid, and it contains the ampicillin resistance gene, and it has the LAC-Z gene in red right here. That's supposed to be this stretch right here, the LAC-Z gene. Then there's also a restriction site right here, and if you manage to open up and cut this plasmid and insert it foreign DNA piece, so you're cutting both the plasmid and the foreign DNA piece with the same restriction enzyme, and then if the foreign DNA piece that you cut out, so you cut out this piece right here, if you were able to insert it right here, it's kind of small, but you can kind of see that orange piece right there. If you're able to disrupt the LAC-Z gene with the insert and everything went successful, then those bacteria that picked up this plasmid, this now recombinant plasmid, they should be able to grow in ampicillin medium because they have the ampicillin resistance. That was here, this light purple stretch right here. So this here is still your ampicillin resistance and it's clearly there. Also, if you were able to disrupt the LAC-Z gene, then these bacteria, so the ones that have this orange piece inserted here, those can no longer produce beta-galactosidase, and they will turn up as blue colonies in this agar right here. So the blue colonies are the ones that you don't want. They just have ampicillin resistance, but there's no insert that ended up going into the plasmid, so no foreign piece of DNA. The white colonies, on the other hand, they had their LAC-Z gene disrupted by some foreign DNA insert, and that means now you need to take a look at those further, because those white colonies, the colonies with foreign DNA, the white ones, those are the ones you would want because you know they have an insert that may be of interest. Other methods for selecting a clone would be colony hybridizations. That means you're using a DNA probe. In the old days, we were all labeling our DNAs with radioactive P32. That's hard beta radiation, not the healthiest method. And nowadays, you can label your probes just as efficiently and with the same sensitivity level with fluorescent probes, which is far less dangerous and also creates no radioactive waste or other dangerous waste. So you have to have a probe. It's kind of like going fishing. You have your hook there and you put a worm on it, and then you go fishing and you hope you're going to catch something. So in a sense, colony hybridization is the same thing. You got to have a DNA probe. So that means you have to have a stretch of DNA that's complementary to your desired gene, and then you're going to label that the complementary DNA stretch, your probe, so that you can find it. If it found its target, it will tell you where it is. So here would be an example for this. You're going to do some colony hybridization here using a DNA probe that was labeled with a fluorescent dye. So you have here your fluorescent dye. So this is your hook right there, and you labeled it with a fluorescent dye. So we're starting out up here. We have a master plate with some colony growth, as you can see. You're going to take a nitrocellulose filter and you sort of do a replica of the colonies that ends up on the membrane filter, the nitrocellulose. And then you were just going to break open the cells to release the cellular contents, and including the DNA. So that will be step 3. And then you are going to, and by the way, DNA binds very tightly. Also proteins bind very tightly to nitrocellulose membranes. So you're going to do some further steps right there, but you're ready to come in with your fluorescently labeled probe, and then the probe will find its matching target based on base pairing rules, and if that happened, then you will see that the colony right here where the probe has found its target DNA, those are the ones of interest, and those colonies contain the DNA of interest that you were going for. And here you have actually looks like two colonies that where the probe found its target, and then you go back to your original plate where the live bacteria is still there, because remember, these here are dead on there, and then you pick these two colonies from your plate and grow them further and analyze them further, maybe sequence them. Okay, moving on to making gene products. E. coli is perfect for doing stuff like that. So E. coli, of course, grows easily. It's doesn't have any special growth requirements. You know what you're dealing with. You know its genome, and so E. coli is a very good organism to do gene manipulations with and to have it produce new products. Let's take a look at what some of these products might be. So here they're talking here about genetically modified E. coli that produces gamma interferon, which you need for the immune system functions, human proteins that promote an immune response. I can think of other things such as insulin, human insulin, growth hormone, some other things. So here are some other organisms that you can use to make gene products. Saccharomyces cerevisiae, of course, that's a eukaryotic cell, and it has a much larger genome than bacteria, and so that one is a good choice if you want to have more complex eukaryotic genes expressed. If you can also use plant cells or whole plants that can express eukaryotic genes. Plants are easily grown, of course. It's low cost, and nobody whines usually when somebody kills a plant. Mammalian cells, you run typically more into ethical issues, but mammalian cells, of course, they're closer to our cells, and then you can make products for medical uses, but mammalian cells are generally harder to grow. And so here are some therapeutic applications. Gene products such as human insulin, you can make that even in E. coli. And subunit vaccines, that's getting more and more popular. It used to be that we were growing everything in chicken embryos, but nowadays you can make subunit vaccines with genetically modified yeast, for example. Non-pathogenic viruses carrying genes from pathogens, antigens as DNA vaccines. That's another therapeutic application. And gene therapy, which that has made a lot of strides in recent years. We have now new CRISPR-Cas targeting gene therapy approaches, and if we have time, we'll maybe do a lab on this, or I think there's a Labster exercise that we can take a look at. But gene therapy is basically that you are replacing defective genes or missing genes with healthy ones that work appropriately. So here is a list of some pharmaceutical products of recombinant DNA, and this is a pretty long list here actually. So there's quite a few hormones, growth factors, or colony stimulating factors, cervical cancer vaccines. Those are made using recombinant DNA. Erythropoietin, that's a hormone that you need to make more red blood cells. You can use that in the treatment of people that have anemias. Interferons, all three of them, alpha, beta, gamma, that work with the immune system. The hepatitis B vaccine, human growth hormone, human insulin, we talked about that already. The flu vaccine, I think it's still mostly made in chicken embryos, but I think they might use recombinant DNA technology these days. Interleukins, let's see what else would be interesting for Taxol, that is a cancer treatment drug. Some vet use examples are listed down here too. So okay, let's move on to therapeutic applications. Using our knowledge from DNA technology, we can engage in things like gene silencing, where you would be silence a gene that expresses a protein that is maybe harmful, such as like a cancer gene. And the way it works is you're using small interfering RNAs, siRNAs, that bind to messenger RNA. That is then destroyed by a RISC RNA-induced silencing complex. So basically, I think it's easier to look at a picture on this gene silencing, the way this works here. So here is your DNA. Maybe this is a cancer cell, and an abnormal gene is here produced from a virus or maybe a cancer cell. And now here's your RNA transcript. It gets processed by splicing and then goes into the cytoplasm as messenger RNA, where you normally would then translate it into protein. But now here comes your siRNA, so the small interfering RNAs. They bind to that messenger RNA, and then RISC breaks down the RNA complex, and so therefore no protein products are being made. Now we're moving on to genome projects. That was a really hot thing for a long time when sequencing first became available, and there were mostly people were using a shotgun sequencing approach. So the idea here is for a genome project that you want to have the entire genetic information of an organism, so its entire genome. The Human Genome Project lasted for about 20 years, and then finally in 2002 it was published, and ever since sequencing has become a lot faster, more efficient, and now we can sequence whole genomes not in 20 years but in a matter of a couple days, maybe a couple weeks or months if it's a really complex one. But let's kind of go back to this shotgun sequencing approach. It's kind of what the name says. So you're basically shooting at the genome and shattering it, breaking it into pieces. So you're smashing the DNA, shattering it into thousands and thousands of pieces. And then randomly you're cloning these pieces randomly into cloning vectors, and then sequencing each one bit by bit, one after the other. And then you are trying to find overlapping sequence portions and trying to see if you can fit the pieces back together, so that you then can assemble this whole genome, and you usually need a computer to help you with that. Metagenomics is the study of genetic material directly from environmental samples, but here we're back with the Human Genome Project, and that was definitely done with the shotgun sequencing approach. It took 2 decades, and there were 2 competing groups that actually were trying to figure this out, Craig Venter and then also the Landers government group, sponsored group. They arrived simultaneously at the North Pole, basically, and both put their flags on there. I'm, of course, using an analogy, but so they both arrived at this human genome sequence simultaneously, published it, one published in the journal Nature, the other one in the journal Science, and they both got credit for it. And basically we should take a look at it, the shotgun sequencing approach. So here would be the entire genome of an organism. Here they're using a bacterium, and you just break that genome, the entire genetic information of that organism. You're just breaking it into random pieces, just shattering it. And then you randomly clone these bits and pieces into a cloning vector. Then you are transforming bacteria with these vectors that are now recombinant DNA, and multiplying these pieces, and then you can sequence each purple piece, basically. And then you need to somehow assemble the pieces to see which one fits together, how do they belong together, and that is usually done with the help of a computer. So here are some more scientific applications. The whole field of bioinformatics sort of came about because the sequencing information that became available just became so vast. So people started to put their bits and pieces of sequence information into a large database, and the databases, they grew and swelled up and just really became huge. And so you really need good processing capability to process all of this information, aligning sequences, finding similarities and percent identities to other species and whatnot. So bioinformatics is a huge field these days. Proteomics is the field where you are looking at the proteins that are expressed in a cell and analyze that. And reverse genetics is sequence a gene first, and then you discover the gene function from that genetic sequence.